In the next few slides, we will talk about the steps of designing a clinical trial. We will first have the aim of the trial and have the rules of patients' eligibility for the enrollment for the trial. We will define the interventions and the endpoint for the clinical trial, and we will estimate the sample size. After that, we will set up the control groups and allocate the subject to the treatment groups. And then, in the end, we will collect the data and analyze the data. The aim of this clinical trial is to determine the absolute value or the relative value of the therapeutic intervention of interest. For example, let's take the clinical trial for the retinopathy in the diabetes patients. So then, in this trial, the patients are insulin-dependent diabetes patients, and the intervention, including the intensive therapy, admitted either with an external insulin pump or 3 or more daily insulin injections, while the comparator was with 1 or 2 daily insulin injections, and the outcome was retinopathy. Secondly, we will define the rules of patients eligible for the enrollment, or who are the patients that is eligible for our study, which needs the criteria, including the clearly defined criteria of enrollment and exclusion. We will include those patients that will get cured in the trial. That means the patients will benefit from this clinical trial. And we will also choose a population of patients that with high incidence of the disease outcome, so that the signs and the symptoms are very obvious for them. And we will also need consider some of the safety issues for the patient. So usually no children and pregnant ladies will be involved in these clinical trials. We will also consider who would like to perform this study. That means we will need a population of patients with high compliance. So let's again take the example of the diabetes retinopathy trial as an example. The eligible and the exclusion criteria that clearly stated in their study means that the major criteria for eligibility, including the insulin dependence as evidenced by deficient C-peptide secretion in the age of 13 to 39 years old, and the absence of hypertension, hypercholesterolemia, and severe diabetic complications or medical conditions, as well as 1 to 5 years duration of this diabetes and no retinopathy as detected by the 7-field stereoscopic fundus photography. This character of the entry criteria and demographic characteristic of the study's interest will provide the readers with an adequate description of this study group. The next is to define the intervention and the endpoint for this study. The intervention definition for the control and treatment should be recorded according to our design for the study. The treatment group was usually the drug or therapy that you want to test, while the control group was the therapy was usually used in the current clinical practice, or sometimes that is a placebo. And the endpoint will measure the improvement or design effect and possible side effect in both the control and treatment groups. And the endpoint is also named as the outcome of the clinical trial. It is to assess the result, and the investigators can compare the rate of disease or the rate of death or recovery or other appropriate outcomes. And the outcome of interest must be measured in a comparable manner in both the intervention and the control condition of this trial to evaluate its efficacy. For example, to assess the treatment efficacy, more than one endpoint can be measured. There are types of endpoints include the measurement of the quality of life, the length of survival, the percentage of patients surviving, the complication rate, the intermediate, and some of the intermediate endpoints that can predictive of the survival or quality of life. For example, in assessing the superiority of the treatment of diabetes, possible outcome measure including the percentage of patient survival at a special time following the initiation of the treatment, or the patient's ability to maintain an active lifestyle, or having a major complication from the treatment. In this slide, we will talk about the sample size for these clinical trials. The number of the subjects to be enrolled in a clinical trial was named as the sample size for this clinical trial, and it must be determined in relation to the primary health research endpoint. The factors that affect this sample size requirements mainly include 3 factors: the type 1 error, type 2 error, and the expected magnitude of the difference in outcome between these groups. Besides this, we will also consider that whether this is a 2-side or 1-side test, and we will also considering the rates of compliance and the number of groups that are involved in this experiment. Here we have the two equations that showing how to calculate the sample size. For the qualitative data, we use the upside formula, in which PC and PE are the incidence of the one condition in control group and treatment group respectively. Z alpha and Z beta are the value of alpha and beta referred in the normal distribution. And the P bar means the average of these two, PC and PE. For the quantitative data, the formula is listed in the below part, while the sigma means the standard error for this, for standard deviation for these two variables, while D means the difference of the two outcome variables. In the next, we will talk about how to set up the control groups for the clinical trials. Randomized clinical trials include a control or comparison group so that outcomes in the treatment group can be compared with those in a group that is not treated. The commonly used control group type in the clinical trial include the standard control, the placebo control, crossover control, mutual control, and self-control. Here we first talk about the standard control. Standard control, also known as the positive control, and it is widely used in the clinical trials. Patients will take the standard treatment of this disease when they are enrolled in our clinical trial. In accordance to this given of treatment is in accordance to the ethical requirement of this clinical study. If an acceptable standard treatment is available, then the control group must be treated with this. It would be unethical to withhold that. And this is compared with the new experimental treatment. It is this feature of an ideal clinical trial that have led to them being called the randomized clinical controlled trial. In practice, it is just a special form of the standard prospective cohort. And the only difference between being that participants are log allocated to the exposed and unexposed group at random. As we know that an offer of the treatment is an offer of hope, and it may bring the expectation of treatment success. These expectations are thought to have a powerful influence on the outcome, so that the use of placebo comparison in a trial is intended to distinguish new treatment that have no more than placebo effect from those who have more of an effect. The placebo effect itself is highly variable, depending on the nature of the outcome and the nature of the treatment. As known as the negative control, the placebo control is given to patients in the control group and is a substance or treatment of no therapeutic value. It must resemble that the test treatment in terms of appearance, shape, taste, smell, and mode of delivery. Commonly used placebos include the inert tables, such as the sugar pills, and some injections like saline, and some of the sham surveys and other procedures. Since the placebos contain no active pharmaceutical components, their use is ethically problematic. The placebo can be applied only when no curative measurement is available or the use of placebo has no effect on the progression of the disease. Another type of the control that was used in the clinical trial is the crossover trial design. It is usually used in the two-period crossover trial. Patients will receive both the treatment, the tested therapeutic intervention, and the control therapeutic intervention in a successive order. For each patient, the order in which treatments are received tested the therapeutic intervention followed by control therapeutic intervention, or vice versa, is determined randomly. A washout period is required between these two periods, with the goal of minimizing the carryover effect, including the pharmacological and psychological effects of the treatment received in the first period to the treatment received in the second period. In this type of trial, comparison can be made within the same patients, and the effect of the order of the treatment can be evaluated. However, this type of trial requires more time, and the less to follow-up rate might be high. The crossover trial design is typically used when the condition being investigated is chronic and the treatment being evaluated for short-term relief of the symptoms rather than to provide a cure. Another one is the self-control. Self-control means that one subject plays two roles. One is control and the other is intervention. It is frequently used in the dermatological studies. In self-control, it tested and the control therapeutic intervention can be compared with the same patients. And this includes the comparison of before or after the tested therapeutic intervention was received, as well as a comparison of test and control therapeutic intervention given on the left or right of the hand, the foot, the eye or limb of the same person. There are other control types, such as the mutual controls, the blank control, and the historical control. As these controls are not randomly selected, sometimes it will introduce bias in the clinical trials. A central tenet of the clinical trial is that the patient should be assigned to different treatment groups by a method that maximizes the probability that the two groups will be similar in the background characteristics, which may influence either the response to the therapy or the primary health outcome measurement. Therefore, the method of choosing for assigning subject to the treatment or control condition of a clinical trial is randomization. And with randomization, the determination of assignment to the treatment group is based on probability alone and is not influenced by the preference of the physician or the patient. The purpose of randomization is to achieve equality of the baseline characteristics of the treatment groups, so that the comparison of treatment is considered fair. If patients have been randomly allocated, it is expected that the groups will be similar in the demographic and prognostic factors. Note that the equality of the groups at baseline is highly dependent on the group size. With very small sample groups, it is unlikely that the groups will be similar in terms of all important variables that could affect the outcome. Random allocation is important because if one group were in some way more ill than the other at the start, this might make one treatment look better or worse, even if there are really no difference. It is because of this aspect of randomized clinical trials, the close similarity of the groups in all respects other than the intervention, that they are generally considered to give the best evidence of all epidemiological studies. The real strength of randomization is that, on average, it will also balance other unknown or unmeasured factors across the groups. There are several methods for the randomization. First, we can use a random number table, which is used in all the time. But currently, we usually use the computer to generate a list of assignments randomly for the controls and for the intervention group. And this randomization often done at the coordinating center for the randomized clinical trial. There are several types of randomization. Simple randomizing is the most straightforward way to allocate patients into different treatment groups that can be accomplished by tossing a coin, or draw a lot, or using a random number table. Here we have shown what is randomization can be performed, as we can see from a random table that we just threw a dial such as this thing, and we can find out which is the number. Another type of randomization is called the stratified randomization. In stratified randomization, patients are categorized first into several homogeneous strata, and according to one factor, such as age, sex, or body mass index, etc., that would be associated with the effect of the therapeutic intervention. Patients in the same strata are then assigned into different groups using the simple randomization group method. It will enable the research to look into separate subgroups to see whether difference still exists in the subgroups. Another type of randomization is blocked randomization, and when sample size is very small and many factors are assigned with the outcome of the therapeutic intervention, block randomization can be applied. In block randomization, patients are divided into blocks, in which with the same number of patients have similar characteristics such as age, sex, disease severity, and risk. Patients in the same block are then randomly allocated into different groups using the simple randomization approach. Here we can list the difference between a stratified randomization and a blocked randomization. So for both stratified and blocked randomization, they control the lurking variables for the study, and they form the similar groups for a control and for the intervention. However, in stratified random samples, it was done when taking the sample from the population and to make them into several strata according to their features. While in blocked design, it is usually assigned the unit into different type of treatment. The next slide, we will talk about the data collection in the randomized clinical trials. So, in clinical trials, there are several information that need to the collector in the trials. First is the baseline data. And the baseline data, namely the data collected when the patients entered a clinical trial, often compares some demographic characteristics, some clinical characteristics, drug use. Data means the previous and present drug use. We will also ask about the behavior and some lifestyle data and the personal and family history of the disorders. We will also collect some of the physical and laboratory test data and imaging data. Baseline data is collected to know the basic characteristics of the patients included in the study. It will provide virtual information about the generalizability of the clinical trial. Furthermore, baseline data between the intervention group and the control group can be compared to verify whether randomization has achieved reasonable comparativity between the two groups in terms of the confounding factors associated with the concerned outcome of the clinical trial. During the follow-up time, the following data should also be collected, especially for the treatment data. We will collect what treatment did the subject actually take, and we will collect the information of the treatment assigned to the patient as well as their actually received treatment. The treatment compliance was referred to the extent to which the patient has complied with the allocated treatment. So detailed information about treatment, such as the dosage, the frequency, the route of administration, and the time of the administration will also be collected. It is common for a patient to change or discontinue his or her assigned treatment for objective or subjective reason. Since some of the confounding factors might be time-varying covariates, attention should be also paid to the change in the confounding factors. In the end, we need to collect the outcome information for the clinical trial. Outcomes are any effect of the test interventions. These effects could be categorized into two aspects, the efficacy and the safety. Several points should be noted when outcomes are determined. First, based on the consideration from various people involved in the clinical trial, multiple outcomes should be selected to comprehensively assess the effect of the test intervention. For example, the investigator might be more concerned about the improvement in the clinical symptoms and the laboratory test result, while patients might pay more attention to the physical functions. In general, a few primary or multiple secondary outcomes are determined in this clinical trial. And second, the quantitative and objective variables are preferable to qualitative and subjective ones. Finally, measurement of the outcomes should be defined in advance and should be kept consistent throughout the entire process. In the next slides, we'll talk about the statistical issues in the clinical trials. First, we have already talked about the sample size for the clinical trials that was determined by the primary study outcome, and it can need to account for the multiple comparison problems. And for the sample size, we may sometimes need to require some statistical consultant to define a good and efficient sample size for the study. And after this monitoring of the outcomes, there are some of the study analysis that can be done when the intern of the clinical trial is finished. So when the clinical trial finishes the intern outcome, the intern analysis can be performed to determine whether to stop the study earlier. And this sample size must be adjusted for the number of the intern analysis. And then we can also consult to the Data Safety Monitoring Board to review these study designs and the study analysis. And for the details of the study analysis for the clinical trials, we should first check the integrity and the validity of the data. And we then will make a statistical description of the data that we are collected. And we will need to draw a flow chart or flow diagram of this process, and thereafter to do the statistical invariance. So the fundamental measurement of the association for the intervention and the studied outcome includes some of the relative risk and the absolute risk difference. For the relative risk, we can consider as the efficacy result. That can be calculated using the relative risk reduction. And relative risk reduction is defined by that using the incidence in the placebo to minus the incidence of the disease outcome in the treatment group and divided by the placebo group. For example, in the previously mentioned clinical trial for the therapy for the diabetes, we can see that we have a standard therapy and an intensive therapy, and in both of these groups, we then compare the retinopathy, the occurrence of the retinopathy in these patients. So we can therefore estimate the risk of the retinopathy in the standard treatment group and the risk of retinopathy in the interventive group, that is the intensive treatment group. And the next step, we can also calculate the relative risk of this association. The other variables that can determine whether a treatment is good or worse is the absolute risk reduction. The absolute risk reduction means the absolute difference in the rate of the events between the two groups. It means that how many people would be benefited from the treatment out of those receiving it. Absolute risk reduction can be calculated by using the rate in the patient group and minus the rate in the intervention group. And we can also calculate the number needed to treatment that was closely related to the absolute risk reduction. It means the number of patients over a given time period that one would need to treat to achieve one additional study endpoint. It can be calculated using that 1 divided by the absolute risk reduction. In addition, it should also be noticed that some of the patients might be lost to follow-up or don't comply with the assigned treatment during the trial, and thus intention to treat analysis or protocol analysis could be applied. In the intention to treat analysis, we will call it ITT analysis. All the patients are analyzed and outcome measurements are compared between the different treatment groups based on the group to which the patients are originally randomly allocated, regardless of whether they complete the trial or comply with the assigned treatment. The ITT analysis usually leads to underestimating the true effect of the therapeutic intervention compared with the control, because it includes patients who are non-adherent to or derived from the protocol. Another type of the treatment is the protocol analysis. We usually call it the PP analysis. The PP analysis, sometimes known as the on-treatment analysis, uses only data from patients who adhere to the assigned treatment. Thus, patients who do not comply with the assigned treatment are excluded from the PP analysis, and this usually results in overestimating the true effect of the tested therapeutic intervention compared with the protocol. Of note, a confounding bias might occur when PP analysis is conducted, since patients who do not comply with their allocated treatment protocol are excluded. The original comparability of the treatment group in baseline characteristics achieved after the randomization process may not be maintained. In conclusion, ITT and PP analysis should be done simultaneously to reach a solid conclusion based on the comparison of results derived from these two different strategies.